So in this video, we're going to be talking about DNA profiling. In the course of this video, we are going to learn about the interpretations of DNA profiles and the uses of DNA profiles. What I want you to understand is that DNA profiling is a method that is done by comparing the DNA profiles of different people to determine their compatibility. So, a DNA profile can be defined as a pattern produced on an X-ray film. This pattern consists of lines which are of different lengths and thicknesses. As you can see here, this one is thicker than this one. And different positions. As you can see, these bars are at different positions. All individuals except identical twins have unique DNA profile. DNA profiles are used to prove paternity and maternity, biological parents. They are also used to determine the probability or causes of genetic defects. DNA profiles are used to establish compatibility of tissue types for organ transplant. What this basically means is that before a donor is allowed to donate to a recipient, they have to check if the tissue of the organ matches with that of the recipient, so that the organ is not rejected by the body of the recipient. DNA profiles can be used to identify relatives as you are related or not. They can also be used to identify crime suspects in forensic investigations. Meaning that when someone has committed a crime and let's say their blood was left at the crime scene, or a strand of their hair, that can be used, and it can undergo DNA profiling to compare with that of the suspects to determine who actually committed the crime. Yes, and then we go down here. So let's interpret the DNA profiles before. So here we are trying to determine which of the four men is the father of this child. 50% of the child's DNA matches with that of the mother, and the other 50% matches with one of the biological father. So one of these four men has to match 50% of this child's DNA profile. The bars, 50% of the bars have to match with that of the father. So here there are 6 bars, meaning that 3 have to match the mother and the other 3 will match whichever the father is here. So yes, let's determine which 3 match the mother. So when we move like this first bar, it matches. This first bar matches with that of the mother, so 1. Here there's nothing. The mother, there's nothing here. The second bar does not match with one of the mother. The third bar matches with the mother. The fourth bar does not match with the mother. The fifth bar matches with the mother. So, and the last bar does not match with the mother. So let's compare with that of the men. So if we move in a straight line like this, you see already this one matches. These B and C match the second bar of the child, and then we move on to the fourth bar of the child. A, B, and C match the child, and the last bar matches with B. If we look properly down here, which of the men matched with the child the most out of all of these men? As we can clearly see, it's B, meaning that B is the father of this child. Yes, that is our DNA profiling. Determining paternity, that's how you do it. So we move on. So the 50% only applies to paternity. When it's a crime scene, it doesn't necessarily have to be 50%. You can just look at the most bars. It doesn't have to be 50%. So next we are going to look at the advantages and disadvantages of DNA profiling. We are going to start with the advantages first. The first advantage is meaning that we get to determine our biological parents. Yeah. And then the second advantage is tracing lost relatives. We benefit like that. The third advantage is that we get to treat our genetic defects when we find them in the DNA profile. One of the disadvantages of DNA profiling is that it can be inaccurate. Inaccuracy of DNA profiling process leads to errors. The second one is that human error in interpretation of results. This means that scientists can interpret DNA profiles wrong, as in when they're trying to read the bars and their compatibility, something could go wrong. They're messing up with the results. Yes. The third disadvantage is that data may be used to discriminate against an ill person. Let's say scientists are testing and observing the DNA profiles. They may find out that, for example, the person is HIV. They are going to use that against the patient is going to be maybe discriminated against because of the results that are obtained. That's what it means. The next point is that the costly process of DNA profiling may exclude poor communities. As in, you are poor and you cannot afford that, you may be excluded because it's too expensive for you, although you are sick or you care about your health. Yeah. And the next point is that DNA samples may be planted at crime scene, causing for people to be framed. Yes, as you can see here, the disadvantages are more than that of are more than that of the more than the disadvantages. So yeah, I'm not saying that these are all the advantages. Most of the advantages are just basically the uses of DNA profiling. So yeah, pretty much the same thing. Yeah, and then these are the disadvantages. Yes, that is it for today's video. Thank you.
Let's get it. Oh, yeah, yeah.